entering into the Moodle, because I have here some instruction how to do it. And I got many emails. לא היה בעיות. מה? Okay, so let's see if I can help. Yeah. And you couldn't find which one? Ah, you found it. So it's piecing together Alzheimer's disease. Okay? So it's the correct noun? Okay. So you don't need this introduction. Did you have any problem with entering into Moodle? או you're fine. היה לך בעיות להיכנס למודל? או אתה בסדר? כי...
לא בא להיכנס למודל, פשוט לא הבנתי...
מה צריך?
על איזה מקום...
אוקיי, אז אני אראה לך עכשיו, כדי שלא... הסברו לך? אתה פיקס? אוקיי. So...
פשוט לא היה מצוין
שזה המאמר שקוראים...
בסיס ה-piecing together Alzheimer.
Okay. So today we are going to talk about two separate subjects. The first one will be structure and functions of cells of the nervous system. And the second one will be a debt that I owe you from last week, which is the RNA contribution to the human diverse proteomics. As you probably all remember, last week we learned the basic dogma that DNA is subscribed to RNA and the RNA is translated into protein. Today we are going to learn about the role and the so important role of the RNA. But let's start at the beginning. So let's start at cells in the nervous system. I want to talk to you today about three main subgroups of cells that plays the most important role in the brain. The neurons. The neurons are the elements that process and transmit the information within the brain. The second subgroup is the supporting cells, the glia. The glia support physically and chemically the neurons. We'll talk about it later. There are three main categories of glia cells: the astrocytes, the oligodendrocytes, and the microglia. And the last cells, which form the vessel walls of the vessel walls in the brain, which provide chemical isolation from the rest of the body. The name of this isolation is blood-brain barrier, and in short, BBB. Sorry? Of this BBB, the blood-brain barrier, it's a physically barrier between the blood and the rest of the brain. So structures like microorganisms and poisons will be able to enter into the brain. But we'll talk about it later. Okay, so three main categories of cells: the neuron, the supporting cells, and the BBB. Let's start with neurons. So neuron is composed from the cell body, or soma, the dendrites here. Can you see the dendrites? The axon, which is covered with myelin sheath, and terminal buttons at the end. Now let's talk about each one of these parts of the cell. So the soma, the cell body, here in the cycle, and here, is actually include the main machinery that provides life processing of the cell. The nucleus that we learned last week, the mitochondria that we learned last week, the endoplasmatic reticulum, all the parts that provide the energy and and the heredity and the proteins for the cell, for the whole cell. Second, we'll talk about the dendrites. Neurons talk to each other in the nerve. As you see here, there is a cell, which is a neighbor of another cell. And this cell here, on the terminal buttons, can secrete chemicals that gets here to the dendrites of the next cell. So in the dendrites, there are parts called synapses that they feel or sense or understand the message that the nearby, the neighbor nerve sent to them. Okay? So yeah.
Just before I summarize, I think, is everything?
All the important things are inside this presentation.
And the presentation is?
I will upload it. It will be. I will upload it at the beginning of next week. Okay? Okay, so the dendrites are, we can say, the ears or the eyes, or they sense the chemical message that gets from the nearby nerve cell. Then the axon. The axon carries the information from the cell body to the terminal buttons here. The axon is a long cylindrical tube that's covered with myelin. Here at the beginning, just between the cell body, the soma, and the axon, there is a special spot. The name is the axon hillock, which actually decide, we'll see later how, if this nerve will send a message or not to the other cell. The message, the name of the message is action potential, which is a brief electrical and then chemical event that starts here at the axon hillock, and it travels through the axon to the terminal buttons. Okay? So the axon is like a cable. The axon is covered with myelin sheet. The myelin is mostly protein, which isolate the axon from the rest of the fluid of the brain. Okay? And help to conduct the electrical message. And then at the end there are those terminal buttons here. When an action potential, actually the electrical part of the action potential, travel down the axon and reaches those buttons, a chemical called transmitter substance, and because it's in the brain, we call it neurotransmitter, which is a chemical secreted out of those terminal, okay? And the neighbor cells can sense them in the dendrites, okay? Is it clear? Okay. So there are lots of kinds of chemicals, of neurotransmitters in the brain. Some of the most common are dopamine, serotonin. Do you have a question? Okay. Or acetylcholine. I just want to give you some hints. In Alzheimer's disease, it is characterized by death of cholinergic neurons. Cholinergic neurons, they are neurons that secrete acetylcholine. In Parkinson's disease, it is characterized by insufficient formation of dopamine in areas in the midbrain, in the substantia nigra, that I think that Professor Soreq was talking about. So Alzheimer is a cholinergic disease, and Parkinson is dopaminergic disease, okay? כן, לכל תא עצב יש נוירוטרנסמיטר מיוחד, שהוא כימיקל מיוחד, שיכול להיות מופרש מהטרמינל בטנס שלו. כל תא הוא יכול לחוש באותו פוטנציאל פעולה, וזה אותו דבר, ופשוט השאלה היא איזה כימיקל יש לו בסוף שהוא יוכל להפריש. אז תא שהכימיקל שלו בסוף הוא אצטילכולין, הוא תא חולינרגי. ותא שהכימיקל שלו בסוף הוא דופמין, הוא דופמינרגי. וכך הלאה. we can compare those networks of cholinergic and dopaminergic and serotonergic to like cellular phones. There is the orange and the telephone and the cellcom, but also they can communicate between each other, and you will learn about it later in the later on classes, okay? But for now, let's say that there is a pathway of cholinergic messages or dopaminergic messages and so on. Okay, so now we are talking about the neurons, and I want to talk to you now about some of the supporting cells. So the name of the supporting cells are glia cells, and they are called glial because at the beginning, researchers thought that they are like nerve glue, that they glue all the nerves together. Now we know that they have much more things that they can do, and they are so important. So if we are talking about all the supporting cells as a group, so they are all surround neurons and hold them in place physically. They control the supplies of chemicals they need. They isolate them from one another, so the message will be clear. They destroy and remove the remains of neurons that are killed by injury or die. We will talk now about the three most important types of glial cell, which are astrocytes, oligodendrocytes, and microglia. So the astrocytes, as you can see here, they look like stars, and they have arms, processes, that they supply physical support to nerve cells, here, and also to blood vessels here. They put everything in place. Also, they clean up waste within the brain. They provide chemicals and nutrients for the neurons, and they help control the chemical composition of the fluid surrounding the neurons by actively or taking out or releasing substances to control the concentrations of those chemicals. Yeah.
What happens if the dead cells are not destroyed?
If the neurons?
Yeah, if the dead neurons. Sorry.
Okay. So I can answer you this question from several aspects. First, when there is a dead cell in a place, it secreted all kinds of chemicals that can induce the death of other cells. So it can enlarge the place of the injury. Also, if one neuron communicates with another, and in the middle there is one neuron that is dead, the pathway is cut in the middle, and there will be a problem with the communication transfer.
And physically, what happens?
How can they destroy?
Outside the brain. I mean, what disease is it?
מה קורה מחוץ למוח?
Yeah, can it create some sort of disease?
If there are problems in supporting cells, it has major influence on neurodegenerative balance, state, degradation, enhancement, and you will talk about it מאוד בהרחבה in the next lessons. Yeah, the astrocyte is very, very important. There is also, I want to answer you in a third manner, that neurons do not generate, usually. If a neuron dies, so it dies, and there is no one that can replace him. So this is another so important feature of the astrocytes, to get rid of the dead. Okay, oligodendrocytes. The oligodendrocytes, you can see it here. It's not my stain, but it's a beautiful stain, I have to say. The green is the oligodendrocyte, and the red are neurons. The oligodendrocytes, it's actually the supplier and provider of the myelin sheath. The myelin sheath composed from 80% lipids. Lipids are fat, and 20% protein. Given oligodendrocytes produce several segments of myelin, okay, that cover the axon. So if now you already know that the axon is like a cable that transfers the electricity, and you know that myelin is composed from lipid. What do you think that the importance of the myelin? What can be the importance of myelin?
Like electricals, you have to wrap them around in plastic sheets.
Exactly. I just want to convince you that the myelin is important. Have you heard about the neurodegenerative disease, multiple sclerosis? So multiple sclerosis actually is a disease that is caused because of problem, because of damage and scar inside in the myelin, and it causes the electrical signal to decrease, not to get at the terminal buttons, and the transform information to stop and not to be efficient. And this is all about the multiple sclerosis. So the multiple sclerosis is an inflammatory disease which the fatty myelin sheath around the axon of the brain and spinal cord are damaged. They get scarred, and it created a broad spectrum of cognitive and neurological problems. And it's just because of the myelin.
MS, ברשת נפוצה?
כן. Okay. לא, אינפלמטורי זה של המערכת החיסונית. אוקיי. כן. Okay. So this was just in סוגריים to convince you that the myelin is important. So the oligodendrocytes are the supporting cells that create the myelin. You can see here a scheme of an axon which covered with myelin, and here an axon of multiple sclerosis patient, let's say, but because it's a stream, that is damaged. Okay, and here the wire is open, and the action potential can decrease. And the last subtype of supporting cell, the bear, is the microglia. The microglia is the representative of the immune system in the brain. They act like phagocytes. They engulf, breaking down, and digest dead and dying neurons, also as the astrocytes. They serve as one of the representatives, as I said, of the immune system in the brain. They're protecting the brain from invading microorganisms, and they're responsible for the inflammatory reaction response in the brain. Okay? And in response to the brain damage. Okay, so we have the astrocytes, the oligodendrocytes, and the microglia. They play a very important role in neurodegeneration. We'll see. And now, I want to describe you an experiment that was conducted in 1985 by Paul Ehrlich. And at the end, I want to ask you, what do you think the significance and what is the main finding of this experiment? So what Paul Ehrlich did, that he injected blue dye into an animal's bloodstream. What happened? That all tissues except the brain and the spinal cord turned to blue. However, the brain and the spinal cord stayed in their didn't get stained. However, if the same dye was injected into the ventricles, ventricles are the rooms within the brain and the spinal cord became blue, but the rest of the body didn't.
What you said before about those blocks.
The brain-blood barrier, exactly. Exactly. So.
You got it, but I didn't.
How can prove it?
Yes.
So he saw, okay. So he saw that when he injects blue dye in the blood, all the tissues in the body except the brain get blue, but the brain does not.
Okay.
But if it injects this blue dye in the brain and the spinal cord get blue, but all the body stays, it doesn't get stained.
And what
So
does
the-
it do?
So he thought, and now we know that it's right, that there is a blockage, a barrier between the brain and the whole body, and the blood in the body.
Ah, okay. A barrier.
A barrier, מחסום يعني. So this experiment demonstrates that a barrier exists between the blood and the fluids that surround the cells in the brain. This barrier is selectively permeable. Some substances can cross the blood-brain barrier and some others cannot. Do you have any idea who will cross and who will not? What the body wants, what the body needs. Okay, so microorganisms, חיידקים, will not cross because we want to keep the brain privileged from microorganisms, but nutritions will, like glucose and all, proteins will not.
Okay.
Proteins, you know that, as you remember from last week, but this week we'll talk about it also, proteins are translated within each cell. The neurons can create its own proteins.
Ah,
Amino acid will pass, but not proteins. Okay? נמשיך? Okay, so let's see how. So in most of the body, the cells that created the vessel wall do not fit together absolutely tightly. There are tiny gaps between the cells, the endothelial cells that create the vessel walls, okay? And through those tiny gaps, the substances cross the blood to the fluid that surrounds the cell. However, within the brain, here, there are no gaps between those endothelial cells. They are very positioned very tight to each other, and the substances cannot cross this barrier. Okay? Okay.
How does the blood get to the brain?
it's inside.
It's inside the vessels. Blood זורם? תזכירו לי?
So it's like a מסננת?
So it's מסננת all over the body, but it's not מסננת within the brain. Within the brain,
There.
sorry,
This is something that's
okay. Okay, so now, till now we were talking about the three main subgroups of cells in the brain. The neurons, the supporting cells, and the endothelial cells that creates the blood-brain barrier. Now, I want to talk to you about the communication within neuron. How neuron understand, create, and transmit a message. Okay? It's all a matter of charges, of electricity, and I need you to be a little bit in focus now. So, the inside of axon is a little bit more negatively charged than the outside of the axon, okay? The differences in charges are about 70 millivolts. In terms of electricity here, it's tiny, but it's enough for our body. These electrical charge differences between the inside and the outside of the axon is named membrane potential. Okay? Actually, the electrical part of action potential is the electrical message that is conducted down the axon. It consists of a brief change in the membrane potential. This is it. Now we are going to see how. סבבה? איזה חלק?
השלילי והחיובי השפיע, אבל כאילו החלק שהוא עובר, מה בעצם קובע את ה...
בעצם המסר החשמלי עובר לאורך האקסון, והוא בעצם שינויים בהבדלים ובהפרשים בין המטענים מחוץ ובתוך האקסון, ועכשיו אנחנו נראה איך לעומק. זה היה... אוקיי. So, let's go deeper a little bit how membrane potential is created. So, the electrical charge is a result of a balance between two forces, diffusion and electrostatic pressure. Let's talk about diffusion. Diffusion is the movement of molecules from regions of high concentrations to a region of low concentrations. Imagine a cup with a sugar. If you put a sugar in a cup and you will not stir, in few days you will see that the sugar is spread evenly in the glass. This is diffusion. Diffusion, it's okay. The second force is the electrostatic pressure. The fluids, both inside and outside of animal cells, contains a high concentration of dissolved salts. The basic of these fluids are water. It's an aqueous solution, and this is a molecule of water. Water is here you see the O, the oxygen, which have two negative charges. To the oxygen, two hydrogen molecules are linked. Each molecule is positively charged with one positively charged. So if we look at this molecule, it's evenly. It doesn't have any charge because there are two negative charges on the O and two positive charges because of two hydrogen. But, and if we look at it on three-dimensional way, we'll see that the O, the hydrogen, is in one side, and the H, the positive charge atoms are in the other way. So if we put something negatively, so it will get attached to the hydrogen, to the positive part. And if we put something negative, if we put something positive, it will get attached to the negative part, to the oxygen. But if we put something negative, it will get attached and get close to the positive part. Minus the plus.
But it will get close, but it won't connect. Or it will...
Not connect, but it will get closer. The water will stay water. The water will not spread apart. It will stay water. Okay, so if we will insert salt, normal salt, like sodium chloride, NaCl, it's a salt, okay? The salt will break up almost entirely into two positively charged sodium ions, the Na, the sodium, and negatively charged chloride ions, the Cl. Can you see here? Is it clear?
The interaction of salt, NaCl,
Water. With water? Separates the NaCl to Na and Cl, and the salts are separated into ions, into positively and negatively charged ions. Okay, so the four most important main players, ions in our membrane potentials are sodium, potassium, calcium, and chloride. The sodium and the calcium are positively charged, and the potassium and the chloride are negatively charged. As simple as that. Can we go on? Now, as I said before, just as the force of the diffusion moves molecules from a region of high concentration to a region of low concentration, electrostatic pressure moves ions from place to place. cation, which are the positively charged, are pushed away from region with an excess of cation, and anions, which are the negatively charged, are pushed away from region of excess of ions. Okay? Okay. Here we can see the lipid membrane. The lipid membrane is not charged, and that's why it's not get dissolved in water, and it creates a barrier between the intracellular part and the extracellular part. Differences in the concentration of ions on opposite sides of cellular membrane provide a voltage difference called the membrane potential. Okay, this is actually the membrane potential. Now, as we said, the largest contribution usually comes from sodium, you see here, and chloride ions, which have high concentration in the extracellular region, and potassium, and long negatively charged proteins with high concentration within the cell, in the intracellular region. So chloride and sodium are outside the cells, okay? And potassium and negatively charged protein are inside the cell. Yes, please.
Why don't you put both Na plus and Cl minus outside the cell? Does it have a positive charge? It seems like if they were NaCl.
It's not evenly, they are not even.
Oh.
They are not even.
Why are they not?
It's a very good question. It's a matter of ion pumps and ion channels, which in a minute we'll talk about it largely. Okay, so for your question, for my invited question. So the resistance of a pure lipid, of a pure lipid bilayer to the passage of ions across it is very high, but structure embedded in the membrane can greatly enhance ion movements as either actively or passively. The two types of structure that play the largest role are ion channels and ion pumps. Both usually formed from lots of protein molecules. So we'll talk briefly about channels and then a lot about the pumps. So let's start with the pumps. So the pumps are known as ion transporter. They actively transport specific types of ions from one side of the membrane to the other. Sometimes they're using chemical energy, which derived from metabolic processes to do so. Do you remember which organelle in the cell produces the energy for the cell? יפה, mitochondria. I feel that I did something like that. About the channels. So ion channels provide the possibility of ions to pass through the membrane. In most cases, the ion channels is only permeable to a specific type of ion. For example, sodium and potassium, but not chloride and calcium. Okay? Okay. Let's talk a little bit more about the ion channels. So there are protein structures that are embedded in the membrane that allow passage of specific type of ions under specific conditions. The ion channels can be divided into three subtypes: the liquid channel, the ligand-gated channel, and the voltage-dependent channel. The liquid channel are actually the most simple type of channel. Their permeability is more or less constant. The most important leakage channels for us that has the greatest significance in neurons is the potassium and chloride channels. Okay. The other type of channel is the ligand-gated channel. Those are channels that their permeability is greatly increased when some types of chemical ligand bind to the protein structure. Here you can see that here the channel is closed, and when a ligand, here, these little yellow things are attached to it, it's open, and ions can pass. Okay? I want to give you a little clue that on the dendrites of cell, there are a lot of ligand-gated channels. The name of these ligand-gated channels are receptors. It's when a ligand, in our case, it's the neurotransmitter, is the acetylcholine, the dopamine, the serotonin, will attach to it, it will open, and it allows passage of ions inside the cell.
So actually the problem is with the ligand-gated channels when, like for in hormone addictions. For example, in drug abuse, where the dopamine action is enhanced or diminished, it has to do with the ligand channels. It has something to do with that, no?
Also, can? אבל אני צריכה לדעת את הזמנים שלי.
לא, פשוט שאולי יהיה צמוד לזה, כי פשוט
יותר טוב לך? I want to tell you now, even a more dramatic thing, that one of the receptors of acetylcholine, the name is nicotinic receptor, because nicotine get attached to it. Cigarettes, normal cigarettes, and they activate the nicotinic receptors, just normal cigarette smoking. And I'll tell you another thing that I don't want you to quote me, that it was found that cigarette smoking is good for Alzheimer's disease, because Alzheimer's disease has there is death of cholinergic neurons. But if you are already sick in Alzheimer and you are 80 years old, you can start smoking, not before. Okay, this is the message from this class. So we said that one type of ligand-gated channels are the receptors that are on the dendrites, and keep it in mind. The last and very important for us ion channel is the voltage-dependent channel. Here I want to introduce you two very, very intelligent and breakthrough scientists, Hodgkin and Huxley, which discovered the ion channels and the whole action potential. They got a Nobel Prize. So the voltage-dependent channels are channels who their permeability is influenced by the membrane potential. Each member having a particular ion selectively and particularly voltage dependence. Many of them are also time dependent. In other words, they do not respond immediately to voltage change, but only after a delay. So, in our words, if there is a change in the membrane potential, there are some voltage-dependent channels that will open immediately, and some voltage channels that will have a lag, a delay till they will get open, and it has an extremely important role in the membrane potential, that we'll see in a minute. Okay? Now, because I'm a little bit short in time, I will just briefly tell you that there are two ion pumps. One is the sodium-potassium exchange pump, which take outside 3 molecules of sodium and in 2 molecules of potassium. It needs energy to do so because it takes 3 negatively charged molecules and insert 2 negatively charged molecules. It keeps outside the sodium molecules and inside the potassium. And another pump exchanger takes 2 molecules of calcium out and 3 molecules of sodium inside. Okay? Now, don't think that they are contradict each other. because they are not because they are not in the same concentration in the membrane. Okay, so 2 channels, 2 pumps. One take out 3 sodium and 2 bring in 2 potassium, and the second take out 2 calcium and bring in 3 sodium.
So these two kinds of pumps can coexist in
Yeah.
the same.
Yeah, they coexist, but not in the same concentration. Ask me? Sure? Okay, so let's get in the action potential. So the action potential tend to develop at the axon hillock, here, just at the junction between the soma and the axon. We'll see how, but the cell integrate the inhibitory and excitatory messages, information that they get from the environment, from other neurons. They interpret them, integrate them, and decide if to conduct a message, an action potential or not within its axon to the next cell. Okay? So let's see how it happens. So here we can see a graph. The x-axis is the membrane potential. The y-axis is the membrane potential, and the x-axis is time. Okay? And here we can see how membrane potential changes and what happened to it over time. Okay? So let's start. Let's look at point number 1 here. So, as soon as the threshold of excitation is reached, sodium channels in the membrane open and sodium rushes in, and there is influx of positively charged sodium ions, produced a rapid change in the membrane potential from minus 70 to 40. Okay? So, the action potential and the threshold is a matter of none of or all. If there will be less voltage, if there will be less, no sodium, no voltage dependent pumps and channels will open, no sodium will get in, and the membrane potential will not get to plus 40, okay? Just if a certain threshold is achieved, just then all the channels will open and all the sodium will get in, will rush in, okay? And will change the inside of the cell will be much more positive than the outside of the cell.
What is the threshold?
Sorry? What is exactly the number?
Yeah.
I don't remember by heart, and let me get back. You know what? I will give you an answer in the site, in the Moodle. Thank you for the question.
Can you repeat again what is determining what will come in and what will not? The electric voltage?
Yeah. So there are voltage, like we said here, voltage-dependent channels, you see? And when the membrane will get to a certain threshold, which I will tell you exactly what is this threshold after your question, all the channels will open and the sodium will get in. It's after voltage dependent. Now, and actually the sodium that rushes in creates the positively charged membrane potential. Now, if you will see, look at point number 2. Less sensitive voltage-dependent potassium channels, they require greater level of depolarization before they begin to open. So what happened? We said that there are those very fast, immediate opening of sodium channels and very slow opening of potassium channels. Now, the potassium channels get out of the cell, okay? Okay, the sodium is getting in and the potassium is getting out. About 1 millisecond. When the action potential gets its peak, here to the peak, here, here, at plus 40 millivolts, all the sodium channels are getting closed, okay? And no more sodium enters to the cell. But the calcium channel, the potassium channels are still open, so all the potassium is getting out of the cell, and like this, the potential is getting down again, from 40 down, okay? Because no sodium is getting in and lots of potassium is getting out. Both of them are positively charged, okay? Let's look at, this is what we said now in 4. By now, the voltage-dependent potassium channels are open, letting potassium ions move freely outside the cell. This outflow of cation causes the membrane potential to return towards its normal value. And as it does so, the potassium channel begins to close again. Okay? Now, here at 5, the potassium channels close and no more potassium leaves the cell. Okay? Now there is a little bit of accumulation of potassium ions outside the membrane, and this is caused a little bit more a temporary hyperpolarization, okay? But when they diffuse away, the membrane gets to minus 70. And this is the basic of action potential, and if you understood it, so you did the most of today. So is it clear? Okay, so let's see. It's all a matter of play of voltage-dependent ion channels and the sodium and potassium that gets in and out. At the beginning, when the cell gets to the threshold of excitation, immediately all the sodium channels opens and lots of sodium gets in the cell and and it becomes positively charged, the inside. Very, very, very slowly afterwards, the potassium channels get open and potassium leaves the cells. And it means that there are lots of influx of sodium and little bit of outflow of potassium. Then, when it gets to plus 40, the membrane potential, all the sodium channels get closed immediately. So there is no influx of positively charged ions, and the potassium channels keep open, and lots of potassium can leave the cell. That causes the decline in the membrane potential. And this is it. This is the play.
I understood the process, but I didn't understand the result.
Okay. The result is that in that way, here in the axon halo, the cell decides that the threshold arrived, and it creates an electrical signal. The electrical signal now rushes through the axon. We'll see how, very speedily, very rapidly, to the terminal buttons. When the terminal buttons will feel this potential, this electrical potential, it will secrete their neurotransmitters, the dopamine, acetylcholine, serotonin, and the next cell, the neighbor cell, the adjacent cell, will feel those ligands, those neurotransmitters, and the next cell will open channels and create its own action potential. And this is the language of cells. It's chemical, electrical message.
So the whole interaction between cells is balance between cations and anions.
This is not interaction between cells. This is the interaction within the cell. Between cells, we'll see. It's a little bit more complicated, but this is how action potential is formed.
So the whole thing is a matter of balance, of getting in and out of balance.
Yeah, exactly. The whole biology thing is a matter of concentrations and potentials, and yeah. there is no שחור לבן. There is more tendency to do, or less tendency to do. You'll see. So good for us that we know now action potential. So how the action potential is conducted through the axon. So the conduction of action potential is by dance. It's jump from a myelin covered axon to the next. So through the axon, when there is myelin shift, when it's covered with myelin, the electrical message moves passively. It just moves, okay? Because it's isolated by this myelin fatty entity from the environment. Then there is a place which named node of Renvier. Node of Renvier are places on the axon that are not covered by myelin, and they are ion channels there. When action potential gets there, there are voltage dependent channels that can open and can increase more sodium inside and induce more more current, more voltage. So the axon passively conducts the electrical disturbance from the action potential to the next node of Renvier. The disturbance gets smaller, but is still large enough to trigger an action potential at the node. The action potential gets triggered at each node of Renvier, and it passed along the myelin area to the next node. Okay, so it jumped from place to place. And the name of this conduction is saltatory, which is the Latin word for dance. Okay? The action potential is actually dance from a myelin place, from a node to a node. מה זה? בוא נגיד לטינית. בוא נגיד לטינית ונסגור את זה. אתה יכול גם להוסיף את זה אם אתה רוצה. Okay, so as I told you before, the whole matter of this action potential is it has the all or none law. There is no small action potential or a little bit or nearby or close to. There is all or nothing. Or there is action potential or there is not. Moreover, once triggered, it cannot be stopped, except in MS patients, when there is problem with the myelin. But it remains at the same size, without growing, without diminishing. It's the same. This is the first law. It's the all or none. The second law is the rate. For example, we can hear something loud or quiet. We can see something very bright or very dim. The same neuron will sense it, and it will be need to pass this message. So the thing is not by the size of the action potential, but with the rate. It can, the name of, if we want to say that a cell creates action potential, we'll say the nerve fires. Okay? So with a weak, if there is a weak stimulus here, so the nerve will not fire lot of action potentials. And if there is strong stimulus, so there will be lots of firing of this neuron. Okay? Now, for your remark, let's talk about communication between neurons. So, once our neuron decided to conduct our action potential, how the neighbor nerve will sense it. So, neurons communicate by neurotransmitter, as you say, through synapse. Synapse are actually the places where a terminal button is placed very, very nearby dendrite of another cell, and they create a synapse. Here in the synapse, there is here, is the axon, you see, and here is the terminal button. And here is the dendrite of the next cell. So the transmitter substance produces postsynaptic potential. A brief depolarization or hyperpolarization that increase or decrease the rate of firing of the axon of the postsynaptic neurons. I will explain what I just said. So we have those neurotransmitters that's secreted out of our cell. And then the next set has the receptors, as we said, the channels that can open or close because of it's the ligand-gated channels. When a ligand is bound to it, and the ligand is our neurotransmitter, and then it depends which channel is get open. If a cation channel is get open, so positively charged ion will get in, and there will be more chance that our cell will integrate it and it will get to the threshold to conduct the action potential. If a positively charged ion channel will open, then there is less probability that the next cell will decide to fire, to proceed the message. Okay? And this is all about. So neurotransmitters exert their effects on cells by attaching to a particular region of a receptor molecule called the binding site. Now, if we'll talk about the structure of synapse. So synapses actually are junctions between the terminal buttons, the presynaptic membrane, okay, and the end of, at the end of axonal branches of one neuron, and the membrane of neuron that receive the message, the postsynaptic membrane. Synaptic cleft, it's this, okay, in the middle between the two of them, is the gap between the pre and the postsynaptic neurons. It contains the extracellular fluid through which the transmitter substance diffuses. Okay? Terminal button contains synaptic vesicles. Okay? Its small rounded object contains molecules of transmitter substance. The vesicles are found in the greatest numbers around the part of the presynaptic membrane that faces the synaptic cleft, okay, next to the release zone. So here is a terminal button, and I don't want to talk to you about the life process of those vesicles. I just want to tell you it's when an action potential gets to the button, the vesicles that places just in the membrane of the presynaptic will get fused with the membrane of the button and will transmitted out. Okay? Okay. So this is for our neuron. Okay? The one that created the action potential and sent the message and decided to send the message out. Now let's talk about the next neuron. I just want to tell you that each neuron doesn't talk just with another neuron. There is interconnectivity of thousands, of hundreds of thousands, of lots of neurons that each one sends or an inhibitory or excitatory message to the other one, and each one in the axon hillock will have to integrate all the information, the positively and the negatively charges, to decide if he wants to conduct an action potential or not. So let's see how receptor can activate it. So when releasing into the synaptic cleft, neurotransmitter diffusing and attaching to the binding site of postsynaptic receptor. When binding occurs, the postsynaptic receptor open neurotransmitter-dependent ion channels, which permit the passage of specific ions into or out the cells by two different methods. So we'll talk now about ionotropic receptor and metabotropic receptor. The ionotropic receptor are the simplest, so we'll talk about them first. So, when a neurotransmitter-dependent ion channel, which is ligand-dependent ion channels, as we said, that is equipped with its own binding site here. When a molecule of the appropriate neurotransmitter attached to it, it gets opened, okay? And the ions can get in or out. Here it's closed without the ligand, without the neurotransmitter, and here it's open. So, okay.
By ions, do you mean the Na plus that start the activity?
Yeah, it can be the Na plus, but it can be the Cl minus. So it can be or excitatory or inhibitory. It depends which channel will get open and which ion will get in or out. Okay. So the same action potential and the same neurotransmitter can be or inhibitory or excitatory to the other cell. Just the thing that is matter is which channel will be there. תגיד, הרדמת אותך לגמרי? אתה גמור?
לא, זה ממש, אל תזכפי את זה. זה לא מגיע.
סבבה. אוקיי. Okay, the next, a little bit more complicated receptor is the metabotropic receptor. Those receptors do not open or close in the presence of a neurotransmitter, but instead they start a change of chemical events that requires the cells to expend energy. So, when a molecule of neurotransmitter substance binds with its receptor, the receptor activates a G protein. This is the G protein that is located just nearby. This G protein consists from 3 subunits. Here you see, like that, 1, 2, and 3. When it gets activated, the alpha subunit breaks away and gets attached to a different ion channel here. You see? So here the ligand activates the G protein, and the G protein, and when it's activated, the alpha subunit moves here. And just when it moves here, this is the ligand of this ion pump, or ligand-gated ion pump, and then it gets open. Okay? So these are the 2 main types of receptors, the ionotropic and the metabotropic. And
All the materials have the gun.
yeah, and so I'll tell you about the cholinergic system, because this is the system I work about. So as I told you, you remember the nicotinic receptor? So this is ionotropic receptor, and there is the muscarinic receptor, which is the metabotropic receptor, and there are lots of subtypes, and it's so diverse.
השאלה שלי היא, נכון ההבדל ביניהם שאחד מהם יש לו את הכימיקל הזה,
נוירוטרנסמיטר?
אחד מהם תלוי בכימיקל הזה שיש בו את חלקיק אלפא, והשני תלוי במטען של
לא. לא? טוב, לא, אז תני לי להסביר שנייה בעברית. ברגע שזה לא קשור פה במטען בכלל, זה רק ליגן גייטד, לא וולטג' גייטד. פה ביונוטרופי הוא עצמו, כשנקשר עליו, הוא נפתח ונכנסים יונים. זה היונוטרופי. זה הסוג הראשון. הסוג השני, שוב, כשנקשר עליו הנוירוטרנסמיטר לא קורה כלום, פשוט הוא מאקטב G פרוטאין שנמצא לידו, יחידת האלפא של ה-G פרוטאין שנמצאת לידו היא הליגנד של הצ'אנל האמיתי שצריך להיפתח.
פה זה כאילו תהליך, שם זה ישיר?
פה, בדיוק. אף אחד מהם לא תלוי פה במתח, והם תלויים רק בליגנד, רק בנוירוטרנסמיטר.
מה שאני לא מבינה זה שהליגנד הזה נמצא בכל החומרים? כל מה שנכנס לכאן? או
בואי תשאלי אותי שוב, לא הבנתי.
המרכיב הזה של הליגנד נמצא בכל
הנוירוטרנסמיטר הוא זה שמשוחרר מהפרסינפסה בעקבות פוטנציאל פעולה. את זוכרת, הוא זה שהשתחרר כאן, רק כתוצאה מפוטנציאל פעולה, והוא החומר שיצטרך למעשה לתפעל את התא שאחריו. Okay, so we'll repeat again what I said before, and it's important. So postsynaptic potential can be either depolarizing, excitatory, or hyperpolarizing, inhibitory, and it is determined only by the particular type of the ion channel they open, and not by the neurotransmitter itself. So dopaminergic neurotransmitter can potentiate or inhibitory or excitatory. Okay, about the determination of this neurotransmitter in the synaptic cleft. Here, as you see, there is a great influence of one neuron that fires neurotransmitters to the other one. But in order for this message to be clear and clear cut and short, there have to be some mechanism that will take away the neurotransmitters from the synaptic cleft. So postsynaptic potentials are brief depolarizing or hyperpolarization causes by the activation of postsynaptic receptors with molecule of transmitter substance. They are kept brief by two mechanisms, reuptake and enzymatic deactivation. Reuptake is extremely rapid removal of transmitters substance from synaptic cleft by the terminal buttons. The terminal button of the presynaptic, they just take reuptake, they take back the neurotransmitters. And the other mechanism is enzyme deactivation. They are enzymes that destroy the transmitter molecule, that breaks them. In our cholinergic system, there is no uptake at all, and there is just one enzyme. The name is acetylcholine esterase, because it breaks the acetylcholine to acetyl and choline. And in all the other neurotransmitters, there is the reuptake. Just let me tell you that in depression, it was found that there is lack of serotonin in the synapses. And the most common, most given antidepressant is SSRI, is serotonin, is selective serotonin reuptake inhibitor, which inhibit the reuptake of serotonin, which will induce and enhance the concentration of serotonin in the synapses. Okay? This is the most simple mechanism of the most important drug now.
It detects the problem at the source. Right?
Sorry?
So it detects the problem at the source.
Exactly. No, it's not at the source because we don't know why there is lack of serotonin. The source was to cure the reason of the...
The reason there was a lack of serotonin was because it was reuptake.
No. There is lack of serotonin. We don't know why.
Okay.
So we treat them with blocking the reuptake, so there will be more concentrated serotonin in there.
When people go off of antidepressants, by that point, do they have normal levels of
Serotonin?
Yeah. Do those levels increase and we just don't understand why? Or what happens when people go off? I have lots of things that I want to tell you, but some of them you will not understand. Sure.
But there are lots of kinds of depressions. Not all of them is because of serotonin. The one that because of serotonin, and also there is seasonal depression and waves of depression, and sometimes if a patient has enough serotonin and then it declines again, and then you will have to take again SSRIs. But also there are many speculations that serotonin is not the only source, that there is a problem with dopamine or noradrenaline. It's very complicated, and this is, yeah? The reuptake and the enzymatic deactivation, of course. So enzyme deactivation is just an enzyme that is located in the synaptic cleft and breaks apart the neurotransmitter. Yeah, no, the reuptake is also easy. The reuptake is the presynaptic neuron, the one that sent the neurotransmitter, take them back. Because they want the message to be very brief. It's a mechanism to keep a very high concentration of a neurotransmitter for a very short time. They take them again. Both of them are not there. Okay. So let's talk a bit in 2 words about neuronal integration. So excitatory postsynaptic potential increases the likelihood that the postsynaptic neuron will fire. Inhibitory postsynaptic potential decreases this likelihood. The rate at which an axon fires is determined by the relative activity of the excitatory and the inhibitory synapses on the soma. There are also synapses on the soma, not only on the dendrites, and the dendrites of the cell. Neuronal integration is the interaction of the effects of the excitatory and the inhibitory synapses on a particular neuron, okay? And it's very simple. It gets to the threshold or not. Yes or no, all or none, okay? Okay, I will repeat what I said in different words. So here, when several excitatory synapses become active, the depolarizing message is transmitted by means of passive conduction down the dendrites, across the soma, to the base of the axon, to the axon hillock. If the depolarization reaches the threshold, the axon fires. Here, you see? If it reaches this threshold, it fires. If it doesn't, the sodium channels will not get open. If, at the same time, inhibitory synapses also become active, they bring the membrane potential away from the threshold of the excitation and cancel the effect of the excitatory effect. Okay? Okay, so this is our first part of the talk, and I see that I have, זה עד 4, נכון? Okay. I have 20 minutes. סליחה. Okay. And then now I want to talk to you about a whole different mechanism, which is important for the next lessons, which is the contribution of RNA to the diversity of proteins in humans, in animals. Okay? Different subject. Okay, so the basic dogma that we were talking about last week is that DNA within the nucleus carries the genetic information of a cell and consists of thousands of genes. Each gene serves as a recipe to build a protein. When proteins are needed, the corresponding gene here are transcribed to RNA. RNA is first processed. Now we'll talk about this processing. And then after it gets matured, the mature messenger RNA transported out of the cell, and out of the cell in the endoplasmatic reticulum, okay? Who makes the protein? Who remember? Adenine, someone? Ribosomes, right. The ribosomes create the protein. Now, let's talk about a gene, a DNA gene. So, the DNA gene has two main parts, the promoter and the coding gene. Coding sequence. Okay, the promoter sequence and the coding sequence. The promoter sequence is a regulatory region. It provides the position of the transcription machinery have to come and start to transcribe this gene. Also, there are strong promoters and weak promoters. If there are lots of sequences that brings in the transcription machinery that enhance them, there will be more chance that this gene will subscribe easily and very fast. However, if there are places of repressors, there are proteins that come and block this machinery and do not allow the machinery to work. Okay? So the promoter is a regulatory region. The coding sequence contains exons, the red, which are separated by introns. The mature mRNA, the coding sequence of the RNA is composed only with exons. The intron will splice out, okay? There are mainly two extra non-coding regions within the first and the last intron, which are regulatory also for translation. So, from the whole gene, just the red parts will be translated into protein, okay? The promoter is a regulatory place, and also the introns. Now, let's talk about the pre-mRNA, the mRNA that get translated inside the nucleus. So, it occurs exclusively in the nucleus. Only after those events are completed, the mRNA export to the endoplasmatic reticulum. The rate is about 40 nucleotides per second, and it means the normal gene will be transcribed in 5 minutes. And also there are two modifications. There is 3' poly-A tail and a cap on a 5' end, I will show you, and this is for stability. And after those modifications at the 3' and the 5' of the gene, there will be splicing to take the introns out and to keep only the exons. The mRNA is relatively stable with a half-life of about between 4 and 24 hours. Half-life is a term in biology that describes the תוחלת חיים, the life expectancy of half of the molecules that exist. Okay, this is the half-life of a molecule. So let's see. So if there is here the DNA genome, here in blue are the introns, and in orange are the exons. So primary RNA transcript is transcribe everything. Then it added the 5' cap, the poly-A tail at the end. Then there is the splicing out, the removal of the introns, and the mature RNA goes out of the nucleus just with the exons and the 5' cap and 3' poly-A tail, okay? Which stabilize the RNA. Okay. Now I have a question. Which creature do you think that has the most gene on Earth?
What?
הכי הרבה גנים. אדם. יש למישהו עוד רעיון? Prof. So let me show you the gene number here, and look at the man. The human has a little bit more genes than a mouse, but much less genes than grapevine or rice.
So they're genetically superior to us?
So this is now what I want to tell you. We definitely don't have the הכי הרבה genes, but what we have are very, very sufficient and diverse mechanisms that can regulate and can expand the number of proteins that we can produce from each gene. Okay? So now I want to take the dogma that I was talking about at the beginning of this talk. The DNA is get transcribed to RNA, and the RNA is get transcribed to protein, and there is one gene for one RNA for one protein. It is not true at all. There are lots and many types of RNA that can get transcribed from each DNA, and there are some proteins that can get transcribed from each RNA. And I want to talk to you now about two mechanisms that are important for our course. Okay? So we are sophisticated, but not in the gene number. So one mechanism that is most important for us is alternative splicing. As you can see here, there are exons and introns, okay? And the coding sequence is actually built from the exons that are glued together in the process of splicing, okay? Now I want to describe you a mechanism of alternative splicing, which means that there is a possibility to take parts of this coding sequence and combine them differently. Can you see it? And from the same gene, there is a possibility to create numerous proteins.
This is genetical engineering? The whole...
לא, זה מה שאנחנו עושים, זה מה שקורה בגוף שלנו בבסיס קבוע. 95% of our genes is going through alternative splicing. It's something that is natural process in our body. Okay? To diverse, to enlarge, the number of proteins.
No, okay. I thought that you were talking about like combining differently the genes to create something.
Within the body, it's not.
No. It's a natural mechanism that our body uses, okay, to diverse its number of proteins. So the alternative splicing, it's a process by which the exons of the RNA are reconnected in multiple ways during RNA splicing. The resulting different mRNA may be translated into different protein isoform. Thus, a single gene can code for multiple proteins. Okay? At the beginning, the researchers thought that it's really unique process, but now we know that 95% of our genome is going through alternative splicing. This is the common, it's not the unique. There are lots of kinds and modes of alternative splicing. The most common is exon skipping. If there is here an exon, so the most common is just to skip one and to take it out of the amino acid chain of the protein. Here we see an example. of 4 exons, you see, of a gene. This is exon number 1, number 2, number 3, and number 4 in the pre-mRNA. And here there are alternative splicing. There is combination in 1, 2, and 4. And here there is combination on 1, 3, and 4. And 2 different proteins are created. And the sky are the limits, okay? So this is the first process that is very, very important in degenerative diseases. What Mona talked to you about, I think in the first Alzheimer lecture, was there is problem in the alternative splicing machinery that that in our lab we found out in Alzheimer disease, and this is it, okay? The alternative splicing machinery we saw that something is not functioning כמו שצריך לתפקד. Okay. Another mechanism that I wanted to talk to you is here. This is a regulatory mechanism. It's new one. It's known about 10 years. The name of this regulatory mechanism is microRNA. MicroRNAs are short RNA molecules, and those molecules are not get translated to proteins. They're stays RNA forever. They are only 22 nucleotides long. They are very, very short. What they do that after the mRNA is get transcripted, they bind to a complementary sequence, and when RNA has a complementary sequence on it, it gets degraded and died, and cannot get translated into protein. And this is a mean of the body to control the concentration and the number of protein that will get translated from RNA. Because it can create an mRNA, and when it senses that it's too much, it sends the microRNA, the complementary microRNA that induce degradation. And this, yeah. Also, we've seen that the microRNA has lots of implication in lots of diseases states, including neurodegenerative disease. And from next week, which Dr. Sebastian Kadener will come to teach you, he'll start to work and to talk about the topics that we're talking about today and last week. And this is it for today. If any.